test. That works. Yeah, I'll switch it around. It was just for seeing what I'm making on the screen. Well, I see the same thing on my screen, so there's no reason for me to see the screen once it's set up. Okay, so that's right. Tangled up the mic and the mouse. The good thing is, I think the part of this module today is also part of efficient algorithms in some bits. It goes a bit beyond that, but so you can use it as a, a refresher on that part. Maybe some of the others have seen this before. This we already covered. So we're in the suffix tree chapter. We're always building suffix trees of the text. We'll think of them as represented like this, where you have all the edges labeled with all the strings. We should just sometimes keep in mind that what is represented in the computer has this structure. But one thing that is convenient about this is that the tries already have to remember the string depth of nodes. Remember that popped up or came up last time as one piece of information we want to add to a suffix tree. And the thing is, you don't actually need to add anything. It's already there. Because these numbers in the try nodes, they're exactly giving you the next character to compare on the outgoing edges. And that's the same as the number of characters on the path from the root to reach that node. And it can be a long path like here with one character per edge, which you see better here. But it could also be one edge with several characters. For that purpose, it's the same. Then the first thing we tackled was good old string indexing, the exact string matching problem. Essentially traverse down the suffix tree and see what happens. Either you end up somewhere where you fall off the tree, then there's no match, or you end up somewhere where there is a match, and it could either be a leaf, or it could be in the middle of some edge, or it could be at an internal vertex. In either case, you just explore the subtree below, and all the leaves there are starting positions of a match. That was the first and easy part. And then we looked at the first problem where we didn't yet have an efficient solution before starting with suffix trees. That was this longest repeated substring problem. Find this pair I and L such that you have a repetition in the text where the same string occurs twice, and we want the largest L. How can we do that? Did we discuss this part in detail already? I think we did. So let me just refresh it before we'll generalize it slightly. So the idea is that any path in the suffix tree to an internal node, by definition, the internal node has at least two children. So there's a branch, there's some path that is joined, that's some part of the path that's joined, but then it diverges into two different further branches. So that has to be a repeated string because it's the joint start of two suffixes. And then it branches off, so you can't extend it to the right. To find the longest such repeated string, you just find the deepest internal node, and that's it. So if you have the suffix tree already, this problem becomes almost trivial. You just have to check for the string depth of the nodes And remember, these are already there because the implicit representation of the compact tries needs that number anyways. And so you just find a maximally deep string depth deep node. And so after the suffix tree is built, we just need another traversal to do that. We discussed the longest repeated substring problem in a single string last time. Now let's try to come back to the original problem that I motivated this section with, the longest common substring between a collection of strings. And we'll see a key tool that we can apply to suffix trees in general here that is very, very useful in all sorts of problems. What we seem to need, if you compare what we currently looked at, the longest repeated substring of a single string and the longest common substring of a collection of texts, it feels like we're solving almost the same task, just that here it's several strings and here is one string. So we somehow need a way to compare across strings. So you could build the suffix tree for each of the texts, but that doesn't seem to help you find matches between the texts. Somehow we need a way to get that all in a single data structure. Now the suffix tree is strictly defined for a single string, but what we can do is concatenate the strings. And you want to be a little careful so that you don't have mixtures, a suffix of one string and a prefix of another string. These we are not interested in. So we put these extra end of string markers, they're indexed so that they're all different between the different texts. The order doesn't matter, it's arbitrary. So we build one long text by concatenating all the texts and separating them with a unique symbol. And then for that, we build the suffix tree. So that means whenever you start in some text, then there's a $, there's another text, another $, and so on. If you say we're looking at this suffix here, then really once you see the first $, symbol, you can stop, because you know from then on this will never match anywhere else. So this will always be one long edge towards a leaf. Right? As soon as you see this, the path needs to be unique, because this $, symbol occurs nowhere else. It cannot occur again. And there can be no sharing beyond this point. So we will have some below this initial part that is part of a single TI. This will branch off into some leaf, and that will always be the entire rest of the string. What that means is for every suffix and for every suffix of every text, we'll have exactly one leaf, and we can pretend that they're labeled like this. They should really be labeled with I as the starting index in the entire, they're originally labeled with starting indices in this concatenated string, but you can just recompute what the text is where it starts and the position in that text. So we'll ignore how to compute these, you just subtract the cumulative sums. Here's an example for these three texts all over the alphabet ABC. And what you notice is there's a whole bunch of leaves, but I've always stopped after the first $, symbol. So this would normally, if you directly apply the construction, this first one would continue with the whole second text and $2 and the whole third text and $3. But we don't care about this. All we want is the suffix of the first text. Other than that, it's using the same construction. You're just building it for this concatenated text, and superficially rewrite the leaves. And remember, the leaves are not stored as strings anyways. These are just starting indices. So we're just reinterpreting these numbers. So what can we do with this? One thing we can do is the longest common substring problem. That was this original one where Don Knuth at some point conjectured it might not have a linear time solution. But it does, and here it is. You compute this generalized suffix tree that we just introduced. And then for every internal node, you write down what kind of leaves are below this node. Namely, kind means what substring, what subtext, what text do they correspond to. And there might be some internal nodes that only have one type of leaf, they all belong to the same text, or there could be any subset of the strings. So we do that bottom up. For all the leaves, we just write J, because that's the single string that this leaf corresponds to. And then bottom up, when we've done all the children, we take the parent and we just compute the union of the two children. We also need the string defs as before. And then the longest common substring that is common to all k strings is the internal vertex of largest string depth that is labeled with all strings. And that's it. Before we discuss the running time, let's see this example. So here's the same generalized suffix tree from the slide before. And here's this annotation with taking the leaf types. So this leaf $2 here is of type 2. This is of type 3. This is of type 1. So this node already has all strings below it. That's why it has 1, 2, and 3, whereas some others might have just a subset. So among all those that have all 3, that will be some top part of the tree. We find one that has the largest string depth. In this case, this is uniquely this one. It has a 3, everything else is smaller. So BCA is a common substring, common to all 3. And there it is, right? And it's also the longest such shared string. Otherwise, we would have another node. In the code, most of these steps just take linear time in the total length of all strings. In particular, computing the generalized suffix tree is computing a suffix tree on the string of all concatenations. So that takes the sum of all the lengths. And these are also simple traversals that just look at every node in the suffix tree once, and the number of nodes in the suffix tree is at most n. Now this second step, if you do it just as I described it, then at each node we might have to deal with sets of size up to k. So you probably want to represent them maybe as a sorted list, or maybe some bit vector. Either way, you might need to spend as much time as there are different strings there per node. So in particular, if the number of strings is small and they're just very long, then this is overall linear. If k is bigger, then in general the time is k times n in this implementation. Okay, so that's essentially this original problem from Don Knuth solved, and the main ingredient that we needed is the suffix tree. This construction that we did, doing the generalized suffix tree and then annotating nodes by what kind of leaves are below them, is very versatile. You can solve all kinds of similar problems using the same approach. And one, I put this slide in green because it's really more motivated from lab techniques, is to check whether your sample of reads from some DNA sample is contaminated with a DNA sequence that you're not really interested in, but which was accidentally dropped into the tube in the lab. So before we formally model this, let me motivate a bit that this indeed can happen. Here's an article from this journal Science. Where are we? Let's move it to the other screen. Which is about some protein sequences from some mammoth, but more famously from a T-Rex. So 68 million years old, that's what it says here. And it's the story of a scientist who was a student of one of the most famous paleontologists of our time, who worked a lot with dinosaur bones. And she was hoping that a bit of bone from this T-Rex skeleton that they found, and that she accidentally dissolved a bit longer in some demineralization solution than intended, there was some substance left that looked a bit like blood vessels. And so she thought, okay, cool, there might actually be non-bone substances, proteins from this T-Rex left after all those many years. 68 million years. So they sent it through mass spectrometry and applied some reconstruction techniques. Without getting into the details of how that worked, they tried to determine the sequence of the peptides, of the proteins in this sample. Now this caused a lot of stir. You may know Nature and Science as the top journals in the sciences, natural sciences. So to get a paper there, you have to show something surprising, and this certainly made some news at the time. But then there were several reanalyses of the same data and also suggestions of what might have happened. I think it's not completely clear, but here they at least claim that it might well be that the sample contained common laboratory contaminants like soil bacteria, bird-like hemoglobin, and collagen. And in particular at the time, this first paper detected a surprising similarity between the proteins in T-Rex and chicken, which was taken as the first molecular justification that indeed birds are the successors of dinosaurs, which is otherwise not contested, but it was the first molecular such proof. But it turns out that at least the evidence is very shaky. And so it's kind of a real problem. You can easily imagine how we're full of protein, right? Whatever exactly happens in the lab, even if it's just you working on it, that there's some contaminants going in there is very possible. So as the second article said, there's some likely contaminants that often end up in samples like this. I guess if you dig up dinosaur bones, then they're often in soil. So having soil bacteria, peptides in there is something you could expect. So suppose we know some typical contamination sources. And now we check our new reads. In this case, it's formulated for DNA, not protein, but the principle for the abstraction is the same. So we have some reads. These come from some DNA sequencing run. And we have some known sources from a database of what typical, I don't know, in this case, maybe chicken protein looks like. And we'd like to find, for all those reads, only those that don't have a long common substring with any of the contaminants. Because if they do, then maybe we should discard those reads as being a contamination source. But we don't want to, so the whole goal here is not to throw the entire sample away, but just keep the reads that are likely new and not from any of these known sources. And we're saying here common substring because it's enough for us if there's a decent overlap. We know that the reads are not precisely read, and we may have only a snippet of some longer gene. So it's enough if there's a long common substring that we declare this potentially a contaminated read. So how would we do this? It looks, first of all, a little different from the previous problem, but you can cast it in the same framework. Here we built the generalized suffix trees of all those sequences, all the contaminants and all the reads. In practice, this might be a problem to do this in one shot, but we'll ignore this for now. Then we look at one of those reads, PR, this has a common substring with some specifics, so fixing one read and one contaminant sequence. This has a common substring of length L. Well, exactly if there's, think about it, if this is our PR and here's our contaminant, CJ, there must be, if there's a shared subsequence, right? We don't know where it starts. But this means there's a suffix starting here and a suffix starting here of the two sequences that share a common path in the suffix tree. So they're the same up to a point and then they split. So these 2 points here, this corresponds to an internal node in the suffix tree where they branch off. So we can find this by simply looking for internal nodes that are at sufficient string depth. We only care about long shared substrings. And they need to have any leaf coming from these two strings below, then that's a match. We have a long substring between the two. So if we mark all the nodes in T it's just a binary decision, is there any contamination leaf below you? Then you get a flag as a dangerous node. And then for any of these marked nodes, we check if they have a high string depth above L. Then any read that has a leaf below them needs to be removed from the sequence, from the sample. And so if you do this smartly, you can do it in linear time in the total length of all those sequences, which is pretty surprising if you think about it from first principles, because it feels like you have to check every pair, but the cleverness of suffix trees solve it for us. As a last example of this kind, we'll revisit a structure that we've seen a while ago, where we were talking about the sequencing problem for genomes, where we want to take all the reads and assemble them into one long string. There we were trying to do it with perfect overlaps so that you have k-mers, all of the same length, and then there's exactly one character that we're missing here and missing here, and these are all in perfect correspondence. That was the setup at the time. If that is the case, then you can use the De Broglie graph solution we've shown there with Euler cycles, and the whole problem is efficiently solvable. But with inexact reads, we can't expect that this overlap is perfect. And so one option that we have is, instead of having this perfect overlap, we have sequences of, then we might as well have arbitrary lengths. And we just find the longest overlap, where I can put one sequence on top of the other. And if they don't match too well, then this is maybe just a short overlap by a few characters. Whereas if they're almost matching like this, then probably they overlap by quite a bit. That's the idea. That means in the end, we'll build a graph of these sequences. And then there's an edge between them, which has a weight that corresponds to the number of characters that we overlap here, or number of mismatched characters, depending whether you want to minimize or maximize. So if you pick out this example here, say if it goes from here to there, then maybe this is the suffix that you can find as a prefix here. And then we're back to the TSP or Hamiltonian path problem. We try to find a round trip that uses every read only once, and where the total, in this case, the total number of overlapped characters is maximized. Now, that second step is, in theory, a hard problem. What we'll focus on here is computing all those weights, because we have hundreds of millions of reads in a usual full genome sequence run. If we do all pairs of reads and try to find these overlaps, that's never going to finish. This is completely beyond, is out of the question. So how can we compute all these overlaps? Formally, we say we have a bunch of reads, and we're interested in this matrix of overlaps, where this is the length of the longest suffix of one read that overlaps with the prefix of the next read. So in this formulation, the output is already quadratic in size. So you would have to spend quadratic time. What you would want to do in practice is any kind of entries that are below a certain length, you just discard and don't make the edge there. For illustrating how we use suffix trees to do all this overlap computation, let's ignore this part. We'll build the generalized suffix trees over all the reads as before. Now, we have to find somehow, let me show the example first. I think this is a bit easier. Here's the same suffix trees as before. And let's try to look at this T2, this string as the second string. So I'm looking for prefixes of T2. So there's these other sequences that come before, and then there's T2. And I'm interested in the longest prefix here that I can find as a suffix in the other strings. For example, if we look at BCAA, then this edge should have a length 2 because there's this AA that we can match. And similarly for the others. How can we use the suffix tree to find this? Well, the suffix tree contains all the suffixes of the other strings. So these are already in the tree. All I need to find is these prefixes. But the prefixes I get by traversing down this string and stopping at every position, if you like. So we'll take this string, maybe AA, BCA. I'll start at the root and go down A, and then BCA, and then I'm at the leaf. And whenever at this journey, there's one of those green marked leaves, namely those that just have a dollar symbol. That means there's some other suffix of some other string that is essentially done at this point, apart from the end of string marker. So uninterestingly, this is true in the root. I can always have the empty overlap. So there's all of these strings potentially present many times, and I have to find the deepest of them, the longest. So I go down so far for all these, for all three, I've only found an overlap of 0. Amazing. I go down 1 character. Now for 2 and 3, I've now found a longer overlap, namely 1 character, the A. If I go down 1 further step, I find, oh, for 3, you can actually do 2 characters. And indeed, I can also do 2 characters for 2 itself. Maybe it's a bit amusing. No, it's not right. That's not a terminal link. That's not green. I have to stick with the 2 here. I also get a self loop, right? In this way. Of length 1. We're not going to use these in the sequencing application, but you could say the algorithm just computes this for free. And from the other string, clearly because it ends with the C, this is going to have an edge of weight 0, or maybe the edge is not even there. So the algorithm has to do 2 things. It has to label the edges that have just a dollar symbol somehow specially. And then it has to traverse every string from top to bottom and take note of what was the deepest occurrence of each other string on such a green edge. That's what's spelled out here. These green edges are called terminal edges. They're the ones that start directly with a dollar symbol. And we have a shared suffix prefix. A suffix of pi is a prefix of pj. If that traversal of pj in T reaches such a terminal edge, and we want to find the deepest of these. So to do it cleverly, you do a single traversal with each of the reads, and for all the other strings, you keep track of what was the deepest you've seen so far. And then when you reach a leaf, then you output all of these. And you can even do a single traversal of the entire tree, not just for any specific read, but you can do all of the traversals together. Then the overall time is linear plus the output size. Okay. That was, by and large, 5 more or less direct applications of suffix trees. You essentially exploit the fact that all this overlap information is kind of just encoded in the internal nodes. All of these methods kind of come back to this. That's fine. That's great. There's a few things you can do on top of that. And a lot more is in Gus Field's book, which is linked. I'll mostly leave it with that. There's one more application that indirectly uses suffix trees that we'll look at now. In the next section, I want to talk about tandem repeats a bit. And these are under the hood applying suffix trees, but actually not as directly as the previous methods. And that we're only using something that indirectly uses suffix trees. And you've seen that before. We'll come to that. Before we get there, a little bit of background and motivation. And you might ask yourself what a tandem repeat even is. And I'll tell you soon. And for kind of putting it a bit in context is we've talked about building the human's genome. And we've talked about aligning reads to the genome to find these small differences where it mostly matches, but then there's a small mutation which might indicate some diseases. What else is our DNA composed of? I think I mentioned this before. There's actually only a very small fraction of human DNA that encodes for proteins. That is a gene in the classical sense. 1 to 2% overall. It's a bit more if you count these introns that are cut out, but those are kind of arbitrary sequences. I'm sure they're not arbitrary in the strict sense, but they don't code for proteins. They don't have this genetic code. So what's the whole rest doing? The genes in the classic sense are only a part of what is known to be functional coding parts in DNA. It's just that a good other chunk, and we don't exactly know how much. This is, for many parts, the exact function isn't understood, so we don't know exactly where to put the cut. But I think people are fairly certain that there's half of the DNA where we don't see that it does a direct function. I still find that kind of surprising and amusing. The term junk DNA fell out of fashion, but I think it is very pointed and still memorable here. So we have protein coding genes, as I mentioned, that's the 1 to 2%. Then there's regions that code for RNA, which never gets translated to protein. So RNA is not just this copy of the recipe book, but rather is itself fulfilling a function as a bioactive molecule, or even by just stopping some other messenger RNA from being translated. Then there's structural support for the DNA as a molecule itself. This is in particular at the endpoints of the chromosome. Remember, the chromosomes are these wobbly things, often with a cross here. And so there's parts that hold things together, and then there's parts in the middle that keep the structure. And these are special sequences in the DNA that just happen to form a better structure there. That's part of this here. There's still a lot left, right? There's still a lot of the other 3/4. There's fun things in there where at least partially is understood that they don't seem to have a function. There's parts of DNA that are, by the sequence they form, made so that they're cut out by certain enzymes, where the DNA is then joined again, but they're inserted at some other place. So it's a cut and paste part of the DNA. They're called jumping genes, but they're not actually coding for anything. They're just moving around your genome. And each time they do this, they add a tiny sequence of extra DNA where they were inserted as a leftover of the copying or the pasting process. Fun, fun. Then there's, as I mentioned, the introns that are cut out. These are part of the genes, but not part of the protein. And then there's pseudogenes, which probably started out as ordinary genes that were copied by some process. And then one of the copies mutated into dysfunctional and was no longer used. But it was fine because the other copy still served the purpose. And so the pseudogene was just still around. And well, if nobody cleans up, that's what happens. There's a fun species, the lungfish, which are the largest known genome. Maybe they're... that's what happens if you write papers in Word, I guess. If you have a proper version management system, you shouldn't have copy of gene v3 newest 2. Now, all of this is somewhat amusing, and you may ask, when do we come back to tandem repeats? The reason is that it's still not random what happens in this 50% of DNA. Despite the fact we don't know the function or don't see a function, there's structure in there that we can exploit. And in particular, you can use this for DNA fingerprinting. That's the at least old school version of, oh, there's a blood sample on this murder place, and we have a few suspects. Let's try to match their DNA to the blood we found. And that's going to connect to tandem repeats. So let me move to the video there.
Hi, it's Mr. Anderson, and in this podcast I'm going to talk about DNA fingerprinting. We sometimes refer to this as DNA profiling or your genetic fingerprint. And basically it started with this guy, Alec Jeffreys. Basically in his lab he was working with X-ray and looking at DNA, and what he figured out is he could tell a lot about a person by looking at their DNA, quote-unquote, fingerprint. In other words, he could see who they were related to or who they weren't. He could tell paternity, for example. And so he was working at the University of Leicester and basically figured out this whole idea of DNA fingerprinting. This is around 1984. And basically for the next 3 years, all DNA fingerprinting on the planet went through this university. And so eventually it was privatized, and this is everywhere. And it'll probably eventually be replaced by just DNA sequencing all the letters in DNA. But to make it understandable, essentially what we have in a human is we have long linear segments of DNA, but within that we have these genes. And so 99.9% of our DNA in everyone is going to be exactly the same. The genes are going to be the same, but again, you're going to have different copies or alleles of those genes. That's what makes you, you. But if we look into this area in the middle, we used to call this junk DNA, but now we know it's really important in controlling gene expression. We find that there's quite a bit of variability in here, which shouldn't surprise us because the gene, makes the protein, and the protein makes the phenotype, and that's really what natural selection is selecting for or against. But this in the middle can go crazy, and so it does. And so an example of one that we use in DNA fingerprinting is something called short tandem repeats. Originally, we started with something called VNTR, variable number tandem repeats, and you'll find in DNA sequencing that you have all kinds of. So we had STRs, we have VNTRs. Before that, we had restriction, fragment length, polymorphisms, and so there's a bunch of different things that we could look at. But we've kind of moved to this idea of these short tandem repeats. They work great. There's quite a bit of variability in individuals. And so what is it? You basically have letters of DNA that repeat over and 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 over. So sometimes, 50 times it repeats. And so what does that look like? Well, if we have these three individuals, we'll call this Mr. Blonde, Mrs. Red, and then Mr. Mustache. And so if we look at these three people, their genes are going to be the same, but these STRs are going to be different. These single or short tandem repeats are going to be different. You can see that Mr. Blonde has more than Mr. Mustache and less than Mrs. Red. And so if I make that a little bit easier to grasp onto, if I count them out and then remove everything else, what we get is variability between all individuals. Everything else was the same, but we see variability in here. And we can cut these sections out using restriction enzymes, and then we can amplify them using polymerase chain reaction, and then we can separate them using gel electrophoresis. So how does that work? Basically, I'll take the DNA and I'll put them in a little well, and so we're looking down on this, and so this is a agarose gel. I could put Mrs. Red's and then Mr. Mustache. I could put those all in DNA. Basically, I would then turn on the voltage. So there's going to be a positive charge here, negative charge up here. DNA is a negative charge, and so it's going to be pulled towards the positive. And so what's going to happen is those little fragments of DNA are going to migrate. And so what does that allow me to do? It allows me to tell the difference between each of these individuals. And so this is their fingerprint. But you can tell that this is a really bad fingerprint because we've got these two are exactly the same here. And so when they really do DNA profiling, what they do is they generally use 13 different sections like this. And then those 13 sections are each going to be highly variable. And so it's a good way to tell who's who. When would we ever want to do this? Forensics is one reason, and then also in maternity, figuring out who's dad. And so let's talk about the murder. There was a murder that was committed. Somebody was brutally murdered by one of these three suspects, so Mr. Blonde, Mrs. Red, or Mr. Mustache. But they left blood at the scene. And so what I can do is I can grab samples of DNA from each of our suspects, and then I could grab the blood itself, and then I could do DNA fingerprinting on them. So before we separate them, you may think to yourself, which of these looks guilty? Who looks like they're capable of murder? And if we separate them then, using that gel, what we can see is that Mr. Blonde is guilty. In other words, his blood matches up with the crime scene. And so what do I mean by matching up? Well, those single or those short tandem repeats, if we look horizontally, are going to be exactly lined up. And if we were to look at Mr. Blonde's son, we'd find more similarities than we would between the others. And so basically that's DNA profiling, DNA fingerprinting. It's much more sophisticated than that, but again, it's kind of on its way out. We'll eventually replace this with DNA sequencing. In the US, the FBI has started creating this database of DNA, which is a little scary. And basically what they use are 13 different areas within the chromosome or the genome, and then they're looking at those short tandem repeats in there. Now, why do I say that's a little bit scary? I think you really want to protect your DNA because as we learn more and more about genetics, what's going to be found in your DNA? Well, predisposition to Alzheimer's or breast cancer. Any of these things, which your insurance company would love to get a hold of. And so, and it also doesn't answer the idea of Mr. Blonde. Did he really do it? I mean, did the police frame him and then contaminate the blood? So we don't know that. All it does tell us is if we have two samples of DNA, the odds of two people having the same DNA fingerprint are astronomical, unless they're identical twins, but I digress. And I hope that's helpful.
Good. Apart from the little bit of conspiracy feeling at the end, it shows you that you really want to find places where you have these tandem repeats. And so that's what we'll look at. Formally, a tandem repeat is just some occurrence of a string x that's followed by the same string again. As in the example here, there was these 4-letter strings. Now, our assumption is we don't really know how long these are, and they might be long. So given a text of n characters, find all the pairs of starting positions and lengths so that L is the length of this x. And that indeed I have 2 occurrences right next to each other. At first sight, it might seem dubious how suffix trees could help here, because we have the information about all the suffixes in the suffix tree, but repetitions like this XX is not so clear how you would find this. You could try to play games with the indices where you find things, but to be honest, that's not really how we use suffix trees here. An efficient algorithm results via some indirect route from suffix trees. Using something we've already seen and used in the last chapter, the longest common extension data structure. Just as a refresher, you have a string, you pre-process that string, and then afterwards you can answer LCE queries, which are given 2 positions in the text, and you ask how many characters can I simultaneously read from these positions so that I see the same 2 strings. And maybe that is immediately just superficially clear that this ought to be helpful. Because here I'm looking for an extension from I and I plus L. Here I can ask for any kind of extensions. That seems like a good match. And I've already mentioned this fact that we used as a black box, that you can do the pre-processing in linear time and build a linear space data structure so that LCA queries afterwards are constant time. And we'll eventually get to this, and it'll eventually involve suffix trees. We'll come to this soon. The last bits of this black box will take us a bit longer. Now, directly using LCE queries also seems not so clear how to do it, because there's many possible pairs. You could try every combination of starting positions and lengths, and that's going to solve the problem. But it'll always take quadratic time. Now, the number of such pairs could also be quadratic. So in the worst case, you have to spend quadratic time if you want to list all of those pairs. But these are, again, most strings will have very few of these tandem repeats. So we want an algorithm that is producing the output and of course takes the time to produce the output. But apart from that, uses maybe much closer to linear time. That would be useful, and certainly for genomic sequences, you don't expect a ton of these tandem repeats. We're much less than n squared. The solution in this case is indeed a little less efficient than what we've seen so far. It's built on this idea that if we have some seed position h that we fix and some length L that we fix, then we can, at distance L, expect if there is a tandem repeat that somehow overlaps this seed position H, then it has to occur also L positions later again. Now, we don't exactly know where it starts, and we don't want to try out all possible ways. You'll see in a second how we use this seed position to be precisely. But first of all, if L and H are fixed, what you would want to do is check if there's an occurrence of X that's somehow overlapping. But it could be moving a bit left and right. And these are all possible and viable occurrences. Now, we can do this in constant time, check if there are any of these occurrences, using the LCE queries. And in particular, we do this one, the L1, is using the LCE query on the text, starting at position H and H plus L. That gives me precisely this. Now, this may or may not be as big as L. If my H was the beginning of the repeat, then it is L, and then I'm happy. I found the tandem repeat already. In general, my H might not be at the beginning of this X, and then I have to scan left from H and H plus L to also find this matching part. And so for doing that, that's a 1, by the way. Let me write it like this. We kind of want the same as LCE, but to the left. And let's be lazy and just write this as to the left. But of course, you can build this using the same LCE data structure on the reversed string and flipping the indices around. So if you're interested in the longest common extension leftwards from these two positions, you take that string, you flip it around. So now it reads backwards, and now my positions are here, and now I can read to the right as I normally would. So you can rewrite this as LCE on the reversed text, and then something like n minus h, n minus h minus l. So that's what we're going to do. What remains left is I still seem to be forced to do this for every possible h and l. So how can I make progress? And the solution there is we use divide and conquer. In particular, if I have a string, there's only 4 different kinds of tandem repeats that I could have. Let's pick the middle position here as a special position and call it H. I could have a tandem repeat here entirely to the left of H, or here entirely to the right of H. Or I could have one that contains H, and there's 2 ways you could do that. It could be in the first X or in the second X. So one option is this. That's essentially what's drawn here, or you could have some modified version of this where you have H in the second X. That's the 4 types of tandem repeats I could have. And now the trick is that A and B are entirely contained in the left and right half. So I'm just going to find those recursively. And for C and D, I have to work a bit harder, but at least my H is fixed. L is not fixed, so I have to look over all possible values for L. But with a fixed H and a fixed L, I can answer whether these overlap, so whether this L1 here and L2, the important bit is that they touch each other or overlap. And that I can test in constant time. All right, so in its full beauty, that's what the code for this looks like. We have a recursive function where we do this C and D part. That's for checking all the tandem repeats where we have H, the middle position, in the first copy of X. We have to go through all possible lengths of that X. And then it's a bunch of math for computing the right LCE queries. So you have to kind of play this out on an example, but it's not conceptually hard. And whenever those 2 together, when they touch or overlap, when this is at least L, then I know, like in the picture here, there's an overlap. There's a way to put my X here so that from the extensions that I know about, it's covering the full L positions. There's an outer procedure that builds the LCE data structure and calls the recursive method. And the recursive method is built so that it appends all these pairs to some output list. That's pretty much it. If you analyze this, then the non-recursive part in this procedure is essentially building the LCE data structures. And that's linear time using the black box of LCEs that we haven't talked about, but assuming that that works. Then over all iterations or all recursive calls of this recursive procedure, we're going to produce outputs here. And this is something I didn't stress here. If we have a situation like here where L1 and L2, they overlap, so they span more than L positions, then you can actually pick any of the starting positions that are in the overlap, and it's each contributing one tandem repeat, which probably means that there's quite some repetition, so it's not going to be very often the case, because it means that somehow the first and last characters must be the same. But that's the general case. So if these overlap, then for every starting position in this overlap range, you have one tandem repeat. So in the worst case, you could have n squared of these pairs, and then you'll have to pay for it. That's why this says recording all output pairs takes on the order of the number of such pairs time over all the recursive calls combined. But if you take this out, then what's left of the function is a bunch of LCE calls, constant time each. We do n of them in the two loops. So all of this non-recursive part on the right is linear time. That's this bit. And then the recursive function, we have to set up a recurrence relation. It spends linear time in each step, and it does the standard style of merge sort like recurrence. So you'll easily see that it's n log n time, same as merge sort, same kind of recurrence. So the total time is n log n, not linear, but it's only very slightly worse, right? N log n is still efficient, plus the output, which usually will be small. Should we do a short break, or what do you think? Okay, it's nobody on the stream anymore, so it's just us, so we can decide what to do. That's it. Yeah, this again. Yeah, well, it reminds you to take a break, right? It's an hour ago.
And the preliminary slides on the
they're very out of date. Yeah. Yeah, there were some of the bits where I wasn't sure whether I'll include them or not, and some I only added late. I'll have to update them, but I might do it when we're done with the new bit. Yeah, that's right. Yeah, it's a cool story, this DNA's fingerprinting, that is started with this one guy in Leicester, and for 3 years, everyone sent their samples there. So
I never thought how this came not to existence, but how humans like knew about this, but
also, it's kind of surprising, right? In hindsight, maybe you say, okay, interesting, there's these repeated things and they're different in different people, but how do you, in the lab, pick out the right portions? This I'm not, the video didn't give much more details. There were some other sources that said a bit more about this. Maybe the actual repetition helps because you know that it's always going to look like the same sequence, and so you have something that binds to that sequence to kind of filter out that specific piece of DNA. Or there's some enzyme that can cut out this specific piece. But yeah, is this all, the whole workflow predates all of these high throughput sequences by decades. So they must have had very different techniques for amplifying the DNA. that part, I'm sure this is the not so trivial bit, right? Maybe that's the reason why for 3 years he was the only person able to do it. Yeah, maybe some molecular biologist can explain this to me in a bit more detail at some point. Right, let's continue a bit with the computer sciencey part. We've used longest common extensions a few times now, and so far I've always just appealed to your faith in me that there's this black box that magically does it. We'll now peel off one layer of that black box, and I'll present you a solution in terms of another black box. What do longest common extensions have to do with suffix trees? There's got to be something, otherwise they wouldn't be in this chapter. We've used this already a bunch, so I think you don't need the definition another time. How could we possibly answer this kind of query if we have the suffix tree around? And that's a beautiful combinatorial connection that maybe isn't hard to see. Like if you think about these two strings, well, they're the beginning of a suffix of the text. So you can start at I and at J reading the text. And if they're sharing some longest common extension, that's going to be a shared path in the suffix tree up to the point where they eventually branch off because there's an internal node, and then they split into two different continuations of the suffix. This always has to happen because at the very least one of them will end with a dollar, which cannot occur in a different position in the other suffix. So all we're eventually asking is, given two suffixes, which is two leaves in the suffix tree, what's the lowest common ancestor? That's the same thing. And specifically for suffixes, we also have this third acronym, the longest common prefix. So longest common extension is a position at some arbitrary index in the string. Longest common prefix, as the name says, is always a prefix. And given two strings, it's unique what you mean by that. And the LCA, the lowest common ancestor, makes sense in a tree. But with the suffix tree connecting, all of the three things are really the same. Right? Longest common extensions of a text starting at two positions is the same as the longest common prefix of the suffixes starting there. And this you get from, well, the string depth of the lowest common ancestor. So as an example, say we pick these two leaves, 1 and 7. So I guess here my lovely banana ban. 1 is this A. 6, 7 is this A. So I'm looking at these two. Just visually looking, the longest common prefix is going to be 2. They share AN as the common characters, and then one suffix is done, so they have to differ. How do you find this? Well, here are the two leaves. The lowest common ancestor is this guy, and it's at string depth 2. So a simple kind of connection to see, but it will turn out useful for solving longest common extensions, because we can express them in terms of the suffix tree. Now we're left, I mean, there's two black boxes now. We've gone from the single black box of longest common extensions to build the suffix tree, which I haven't told you how to do it. And then we also need to find lowest common ancestors, which we haven't talked about yet. Now you could take two leaves and just walk up the tree step by step until you meet. That's a way to find the lowest common ancestor for sure. If you have to think about it a bit cleverly that you don't miss each other in a way, but that's clearly doable. You can use the string depth, but it might take linear time because you might have to walk all the way up to the root. So that's not going to get us constant time queries for sure. The other extreme, as always with these data structures, that's the lazy version. I'm not going to do anything in the pre-processing and do everything at query time. The other extreme is I'm very eager. I'm preparing all the possible solutions, but that's going to take a lot of space. and to compute all those. The same goes for the pre-processing. So that certainly doesn't give linear time pre-processing. So we need something clever. And that's the second black box, which will take us to another chapter to solve. We'll essentially replace lowest common ancestors by range main queries, and then these will eventually be solved. And then there's no more hidden nasty boxes that are not resolved. So for the moment, we at least got rid of this LCE black box, and from LCE, we now know we can reduce it if we have suffix trees and LCAs. Again, those two I haven't told you, but they're coming up. Let me also say a bit more about suffix trees in practice. As you've seen, they're lovely to construct all sorts of algorithms on paper and whiteboard. If you implement them, the theoretical guarantees are good. If you implement them, even though they're linear space and linear time, the overhead is quite substantial, especially if the alphabet gets bigger. You have to worry about how to represent the edges, the child edges with the labels. And so, by and large, people shy away from using suffix trees more and more because of the overhead that they bring. There's direct methods that indeed construct the suffix tree of a string in linear time. But these, again, they're somewhat complicated, certainly more complicated than what I'll show you, and have many of them have some side remarks and asterisks. Maybe they only work for constant size alphabets and things like that. So overall, suffix trees rather serve as a good abstraction of what we'll do. We're not explicitly storing them just the way I showed you, but what we'll do is design algorithms in the language of suffix trees and then have another layer below them how to implement these operations. It's still the best way for me to build intuition how to solve a string problem. I tend to always think of the suffix tree first because that's easier to work with. But for practical purposes, there's somehow very fat linear time data or linear data structures. And so I suggest we start a bit with this and serve the main method of suffix sorting then for the new year. But at least the first bits up until the simple sorting methods should be plenty time to do it. I'm not sure if we'll ever get to the phylogenetic trees, we'll see. So I mentioned suffix trees are a good abstraction, but we want to replace them in practice with something leaner. What could that be? That's where we start in this section. We'll try to put our suffix trees on a diet. So if you draw the suffix tree in a specific way, and what I mean by that is make sure that the child edges are in alphabetic order. So always the dollar child, if there is one, comes first. That's preceding all the other letters in the alphabet. Then A comes before B, comes before N, and so on. If you do this at every internal node, you'll notice that the leaves, if you read them in the same order, left to right or top down here, they will give you all the suffixes of the text in sorted order. By definition, because the first come from the branch with the dollar that precedes everything. Then come all the strings that start with an A. Well, then they all precede whatever other suffixes there are. But then within all those that start with an A, I'm going to recursively sort by the next character. That's precisely what aligning the suffix tree like this does. Now, how about we just forget about the tree and only use this sorted order of the suffixes? I'll call this the leaves array, L for leaves. Other texts call this the suffix array. That's the same thing. And you'll see in a second that there's another array where it's very easy and inviting to confuse the two. And so my hope is that this is more mnemonic than just calling it suffix array. We'll come to that. As a first kind of sanity check, let's see if throwing away the tree leaves us with anything that's still functional. And as a sanity check, let's do string matching. If we're given a pattern, can we still find all the occurrences of that pattern in the text, even if we don't have access to the tree anymore? And the answer is yes, at a slight slowdown at this point, but we can use the pattern and do a binary search for it in this sorted list. For example, the pattern A and A, I'm going to do basically two searches. One for A and A with the dollar afterwards as the shortest symbol, and then one with an artificial symbol that comes after every other character. I'm going to do these two searches. Now, this is binary search. You know how it works. It would start here in the middle, then you compare. Okay, I have to pick a middle string. Let's pick this one. Is this before or after A and A in the alphabet? It's after. So I'm going to cut things in half up here. Then so far it's like this, right? Then let's compare AN dollar. If we look for ANA dollar, then AN dollar comes before, strictly before. Then I might have this other check. So ANA dollar now comes above. So I'm going to move this up. And then, well, two more steps or so, I find that, clean up a bit, ANA would go here. That's what binary search returns. It's the position in the array where I would have to insert this if I wanted to keep the sorted order. If I do the same thing with the ANA hash, it'll come after all the ANAs, so it'll have to go here. And now the beauty of these two positions is that they precisely delimit the region of suffixes that start with A and A. So with two binary searches, I found all the occurrences, namely all the suffixes where the suffix starts with A and A. And so that's the cost is that of a binary search, but every comparison compares two strings now. And in the worst case, these might be long strings and they might share a lot of characters. So I have to check from the beginning until I find a character where they differ, and then I can decide the comparison. So that's why it could cost up to M character comparisons to do one string comparison. And this distinction between comparing a whole string, which might read a whole long prefix, and comparing a single character will come up in a second again. But for now, let's say this is still very good. Essentially, the text is what is really long, and the patterns we tend to think of as not quite as long. And the dependence on the text is still logarithmic. It's not as good as suffix trees, where this was irrelevant. The text could be as big as you wanted. It was constant in terms of the text. And we only had M steps in the suffix tree traversal. Here is slightly worse, but we're also not storing all this tree nonsense. So all these pointers are gone, and we just need the sorted list. And remember, this is what we tend to write it for humans like, but really the suffix array is just a bunch of numbers, the starting index of the suffix. So even if we just store this single array, the leaf array, where this is the position R gives you the rth suffix in sorted order, you can still do string matching. Okay. Now we found another cool structure that we don't know how to produce at first. How do we compute the leaf array? Clearly, by definition, if I have the suffix tree, I can read off in a traversal the leaves in order. But hey, we're trying to get rid of suffix trees, so let's try to get rid of suffix trees. We can directly do this by sorting, though. It's just the suffixes of t, they're there, and we can just throw any sorting algorithm onto it. That's trivial to code. You literally do that. Right? You sort the integers 1 to n, and as a comparison function, you give it read the strings that start at that position. Is a one-liner in Python. Now, in the worst case, each comparison can be expensive. And so the best we can say as an upper bound, and even in the worst case, it'll be achieved, is n squared log n time to construct the suffix array. That's in a way even worse than the stupid construction of the suffix tree when you insert the suffixes one after the other. So we've managed to make things even worse. But of course we can do better. Number one is we don't need to use a stupid general purpose sorting method. We know what we're doing, we're sorting strings. So let's exploit that fact. So let's spend a few minutes on the general problem. You're given an arbitrary list of strings that you want to sort. I'm stressing this here because the ultimate solution here will be slightly different, because we know it's not just an arbitrary list of strings, it is a very specific list of strings, namely the suffixes of one big text. And that is extra information, right? This is not an arbitrary list of strings in our application, but we can actually get quite far with just exploiting that we're sorting strings here. As we saw, in the worst case, if I just use black box string comparisons, I guess that might be worth saying explicitly again, even though it's mentioned here. Comparisons can be between entire strings or individual characters. What I mean here is entire strings. These can cost a lot of character comparisons. Now, these are costly if and only if the strings share a long prefix. Okay? This is lexicographic sorting. If the strings differ in the first character, I'm checking this one character and I'm done. It's only really a problem if they share long prefixes. Now, whether or not that occurs or not in practice depends what you sort. If you sort URLs in the standard way where you start with the top level domain, then the next, so you kind of reverse the way we usually write the domains, then they might have a lot of common prefixes. Right? Everything that's from the same page, from the same web, from the same host, say, starts with the same top-level domain, then domain, and so on. So it's not atypical that strings that are sorted have some long common prefix. So we should optimize a bit for this. And the key to doing that is to avoid comparing shared prefixes over and over again. It's as simple as that. And there's two main options. As with sorting, merge sort and quick sort are the way to go. You can, in both cases, for merge sort and for quick sort, find ways to make them not stupidly compare prefixes again and again. The tricks to do that are fundamentally different in a way. And I've decided to just show the second one. You could do merge sort, where the merges, everything in merge sort is about the merges, right? The rest is just recursion. Now in the merges, you have to remember the longest common prefix between the current value and the next in the sorted run. And if you do that, then whenever you do the merge, you're picking the minima of the two runs. Then you know what's the common prefix between the current key and the previous one. And you know this for both runs, so that if you take the smaller of the two, you know how many characters you definitely can skip. And so you can also produce these values again in the output run. I'm not going to further look at this. This is just for for your interest. At the level I explained it here, I think it's not part of the exam. If we find a nice excuse to put it in the exercises, maybe that's a way to teach you about it. What I want to show in a bit more detail is the quicksort version that you can extend to sort strings efficiently. There, all you need to do is to have a partition function that looks at a single character, namely the dth in this write-up. So I'm only focusing on the dth position in all the strings. And my pivot is also a single character. I'll pick a pivot string, but I'm only looking at the dth character. So there's three possibilities what the dth symbol of all the input strings and my pivot string are like. They can have a smaller character or the same or a larger character. But we need the three-way split here, unlike standard quicksort where you just have smaller, larger. Here we need all three. Then on the smaller and larger, I do a recursion just as for quicksort. But on those that start with have the same dth character, I'm going to skip that character in the recursive call. So initially I set d to 0. I'm starting with the first position. But then suppose I'm sorting URLs and they are all from org, Wikipedia, whatever. Then the first position, they will all end up in the equal position for the last character, for the first character of the top level domain. And then they're all gonna stay in this equal partition, but I'm advancing d to 1. So the next partition step, I'm gonna find, according to the second the character at position 1, where they are. Maybe they're still all equal, but I don't forget that they're have been checked. I'm not going to look at these characters ever again. Let me show this on an example a bit. Here's some strings. And I highlighted the dth character that we're currently looking at. So initially here's my list, and I pick some pivot string is always going to be the first one in the example. Then I partition according to the first character and s. There's two strings that start with something smaller than s. There's a bunch of things that start with s, and two strings that start with something bigger than s in the alphabet. And you see, for these, I still have to look at the first character. I still might have to sort them, like these are not in order. But for all those that start with an s, I already know they start with an s. I don't need to ever check this again. So I move on to the second character. So here, you pick another pivot. Now you separated the two strings, so it's singled, that's done. Here I pick a pivot again, and now I partition all those strings according to the second position. There's a bunch of characters that have something smaller than h. These all start with an e, but I don't know that yet. All I did is compare with h and found that these are all less than h. There's one bigger than h, and then there's a bunch with h. And for all those, I advance to the next character because I know they start with sh. And that's how recursively you go. Here in this case, you're not going to make much progress in splitting, right? They stay together, it's the same string. But I don't know that yet, so you move through this step by step. Once you have a single partition, you stop. So here is another case where you move through all the strings. But you see, in each case, you either split something up or you advance and make more things gray. You're not going to not make progress. Okay, I'm going to spare you the rest of the example. You can look through it if you want. And here it would take a few more rounds to check all those characters. I want to briefly analyze how many comparisons we need for this, because this seems like a big mess. In particular, it seems to depend on the input strings, how many characters I need to compare. Like here, if I have lots of strings that are the same or have a very long shared prefix, I can't say how many of these rounds I have to do without looking at the specific strings. It's not going to be a function just of n, the number of strings. But it's actually very easy to analyze with a clever trick. We divide the kind of comparisons that we do into two classes depending on the outcome. This you can only do in the analysis, but that's fine. We do the analysis. There's decisive comparisons, which are comparisons that decide a string comparison. So it's a character comparison, looking at two individual characters, where the outcome was strictly less or strictly greater. The reason they're called decisive is that if you were to compare two strings, then there might be a bunch of comparisons, character comparisons that come out as equal, but then once the first one comes out as less or greater, you're done. And they decide the string comparison. So that also means we can have at most one such decisive character comparison per string comparison, if you were to revisit the original algorithm. And this algorithm doesn't look like it does it that way, but if you combine rounds, effectively you're partitioning the strings by SHE to the extent that you have to. So for this partition, you already know they're less than SHE. For this, you know they're bigger than SHE. For these, you don't know yet, but you're going to continue. So eventually, you know that these are SHE with something behind, so these are bigger. These are S and then something not H, so they're smaller. And these are SH and then something bigger than E, so these are bigger. Over the course of potentially several rounds, you will eventually have compared the strings with she. And all of these comparisons, every string with she, over the course of several of these rounds, there will be at most one decisive comparison, exactly one, I guess, between any of these strings and she. It might just be delayed. You might only discover this decisive comparisons later. And when exactly is not clear, but it will be just one. So in a way, you can analyze the decisive comparisons just as in the standard sort where you have comparison-based sorting, forget about strings. It's going to be the same number. And for quicksort, we know how many these are. If you pick random pivots, we know precisely what the expectation is. The other comparisons I called LCP comparisons because they're essentially telling you about a common prefix. They come out as equal, and that means there's two characters at the beginning of some string that are equal. The important bit here is that we'll never ever compare these characters again because the structure of the recursion with this parameter d means that we remember that they're equal. And the way to analyze how many you need there is a clever reformulation of the problem. The LCP comparisons are precisely the steps that you would need if you were to insert all the strings into a trie. So a standard uncompressed trie where you just have nodes for every character that you go down. I'm not saying that's what we do to implement this, right? This is it's just a metaphor for the analysis. If you imagine you would build a trie for these strings, you would start here, then there's maybe an A here, but then you don't need to go further to compare RE. That's no longer needed. The A was enough. So I should maybe say it's the kind of trie where you contract unary paths to a leaf, but you leave other unary paths unchanged. So but it's not a compact trie. Let's do one more example. So there's the B, which only has the bi. Then it gets more interesting with S. There's a lot of S. And now here, there's SE, and there's SH, and there's SU. But then with SU, we already have the surely. Whereas SE is split up further, right? All of these here. There's an A, and there's an L. And if you have twice the same string, that's a bit of a degenerate situation that we probably for the analysis would try to avoid. I think we would, in this case, assume there's L, S, and then there's 2 dummy leaves, one for each copy of the string. So you would build the string like this, and if there's a unary path that further splits at a later point, you would leave it. But otherwise, you would include all the strings. Now, the number of LCP comparisons is precisely the number of edges here that you have to follow to insert a string. And so this maybe doesn't look like it's all that helpful because it still depends on the strings, but it tells you precisely that that's the number. And the good thing about this try is it's the minimal number of such comparisons that you have to spend to even just verify that a given claimed sorted order is correct. If you were given the strings as a list and you were to check, are they in sorted order, you would compare the first and second. Well, you have to compare the first characters for doing that. Then the second and third, at some point you might have cells and cells. We know that they're equal, but the algorithm doesn't, so it has to compare all the characters. And that's precisely these paths down here. Which means that that number of comparisons is needed by any string sorting algorithm. So the number of these is kind of optimal even without doing much, as long as you don't repeatedly compare things. And the other number we can bound by another analysis. Okay, so overall, this is still simple to code. All you do is quicksort with this extra partitioning method where you check a single character. And it works well for typical kind of strings. In particular, if the input are random strings, each character is randomly chosen independently of the others, it's still an efficient algorithm. But it does have the same worst case because if you take this text of all A's, the suffixes have maximally shared prefixes, and the try to build for all these, there's nothing you can leave off. So in the worst case, this is still bad, and there's no hope to do better with any other kind of string sorting method. It's not Quicksort's fault. It's just inherent. As long as you use a general purpose string sorting method, this worst case will remain. In many practical situations, this is already good enough, and you would probably not see anything that is as repetitive as this text, and so probably it's fine. But we'll see, we can do much better. We can do linear time even in the worst case. But not this year. Goodie.